Hey everyone and welcome back to another new video. So in this session that we will be talking about the biosynthesis of purines.
Starting with the introduction, nucleotide consists of a nitrogenous base and a pentose sugar and a phosphate. So the pentose sugar is D-ribose in ribonucleotides of RNA, and while the pentose sugar is deoxyribose in case of DNA. Nucleotide participate in almost all type of biochemical processes, and they are considered to be the structural component of nucleic acids. So now let's discuss the biosynthesis of purine ribonucleotides. So the main components that contribute to the purine ring of the nucleotides are N1 of purine is derived from amino group of aspartate. C2 and C8 has been arise from the formate of N10 formyl tetrahydrofolate, and N3 and N9 are obtained from amide group of glutamine. C4, C5 and N7 are contributed by the amino acid glycine. C6 is directly comes from carbon dioxide. And it should be remembered that the purine bases are not synthesized as such, but they are formed as a ribonucleotides, and the purines are built upon a pre-existing ribose 5-phosphate. So as we already know that the liver is a major site for purine nucleotide synthesis, whereas the erythrocytes, leukocytes and brain cannot produce purines. So here represent the source of individual atoms in purine ring. So now let's look about the reaction in biosynthesis of purine. The first step is ribose 5-phosphate will react with ATP, and that will result in the formation of PRPP, which is phosphoribosyl pyrophosphate. And coming to the second step is glutamine transfer its amide nitrogen to this phosphoribosyl pyrophosphate just to replace the pyrophosphate and produce 5-phosphoribosylamine. The enzyme PRPP, glutamyl amidinotransferase, is controlled by feedback inhibition of nucleotides. So, this reaction is a committed step in purine nucleotide biosynthesis. So, coming to the third step is phosphoribosylamine react with glycine in the presence of ATP to form glycinamide ribosyl 5-phosphate or glycinamide ribotide. Then coming to the fourth step is N10 formyl THF that donate the formyl group, and the product formed is formyl glycinamide ribosyl 5-phosphate. Then coming to the fifth step is glutamine that transfer the second amino group just to produce formyl glycinamide ribosyl 5-phosphate. And the sixth step is the imidazole ring of purine is closed in an ATP dependent reaction to yield 5 amino imidazole ribosyl 5-phosphate. And coming to the seventh step, that is carboxylation, which means incorporation of carbon dioxide occurs, and that may result in the production of amino imidazole carboxylate ribosyl 5-phosphate. And this reaction does not require the vitamin biotin or ATP, which is in the case of most of the carboxylation reactions. So coming to the eighth step is aspartate condenses with the product in reaction 7 to form aminoimidazole 4-succinyl carboxamide ribosyl 5-phosphate. Then, coming to the ninth step, Adenosuccinate lyase cleaves off the fumarate, and only the amino group of aspartate is retained to yield aminoimidazole 4-carboxamide ribosyl 5-phosphate. Then the 10th step is N10 formyl THF that donate one carbon moiety to produce 4-aminoimidazole 4-carboxamide ribosyl 5-phosphate. And coming to the last step is the final reaction is catalyzed by cyclohydrosylase leads to a ring closure with an elimination of water molecule, and the product obtained is IMP from which other purine nucleotides can be synthesized. So next, let's look about the inhibitor of purine synthesis. The structural analog of folic acid are widely used to control cancer, and they inhibit the synthesis of purine nucleotide. So the given pathway that represents the synthesis of AMP and GMP from inosine monophosphate, that is IMP. Okay, from IMP, AMP and GMP has been synthesized. So the details or the explanation of this particular pathway will be given in the next slide. So let's look. The inosine monophosphate is an immediate precursor for the formation of AMP and GMP. Aspartate condenses with IMP in the presence of GTP to produce adenyl succinate, which on cleaves forms AMP. Okay, and for the formation of GMP, IMP undergoes NAD plus dependent dehydrogenation just to form xanthosine monophosphate. And glutamine then transfers amide nitrogen to XMP to produce GMP. So coming to the next topic is, let's look how the formation of purine nucleoside, diphosphates and triphosphates takes place. The nucleoside monophosphate, that is AMP and GMP, have to be converted to its corresponding di and triphosphate to participate in most of the metabolic reactions. So, this is achieved by the transfer of phosphate group from ATP. And this particular process is catalyzed by nucleoside monophosphate kinase and nucleoside diphosphate kinase. So next, let's look the salvage pathway for purine. The free purines, that is adenine, guanine, and hypoxanthine, are formed in the normal turnover of nucleic acid, that is particularly RNA, and also obtained from dietary sources. So the purine can be directly converted to corresponding nucleotides, and this particular process is called as salvage pathway. So here shows a representation for the salvage pathway of purine nucleotide synthesis. So adenine phosphoribosyl transferase, which catalyzes the formation of AMP from adenine. Then hypoxanthine guanine phosphoribosyl transferase, it can be said that HGPRT, that converts guanine and hypoxanthine respectively to GMP and IMP. Then coming to next is phosphoribosyl pyrophosphate as a donor of ribose 5 phosphate in the salvage pathway. And the salvage pathway is particularly important in certain tissues such as erythrocytes and brain, where de novo synthesis of purine nucleotide is not operative. And if there is any defect in the enzyme HGPRT, that causes Lesch-Nyhan syndrome. So next, let's look about the disorder of purine metabolism. The primary thing is that hyperuricemia and gout. So uric acid is the end product of purine metabolism in humans. So the normal concentration in uric acid in the serum of adult is 3 to 7 mg per dl. So in women, it is slightly lower than men. So the daily excretion of uric acid is about 500 to 700 mg. So hyperuricemia, which is referred to as an just the elevation in the serum uric acid concentration. So this is sometimes associated with increased uric acid excretion also. So which can be called as uricosuriia. So next, let's talk about gout. So gout is a metabolic disease that are associated with overproduction of uric acid. So at the physiological pH, the uric acid is found in more soluble form as sodium urate. So in severe hyperuricemia, crystals of sodium urate get deposited in soft tissues, that is mainly in joints. So such deposits are called as tophi. And this causes inflammation in joints that results in a painful gouty arthritis. So, the primary gout is an inborn error of metabolism due to the overproduction of uric acid. And this is mostly related to increased synthesis of purine nucleotides Lesch-Nyhan syndrome that results in primary gout. So the secondary gout is due to various disease causing increased or decreased excretion of uric acid. So the increased degradation of nucleic acid is observed in various cancers. So next, let's look about the treatment of gout. So the drug of choice for the treatment of primary gout is allopurinol, and this is a structural analog of hypoxanthine that competitively inhibit the enzyme xanthine oxidase. So the allopurinol is oxidized to alloxanthine by xanthine oxidase. So the alloxanthine is a more effective inhibitor of xanthine oxidase. So this type of inhibition is mainly referred to as suicide inhibition. So this was all about the biosynthesis of purine. So I hope that this video would help you for your quick revisions. So thanks for your watching and please subscribe on my channel and don't forget to smash the like button, okay? So see you soon and have a great day. Thank you so much.